Welcome to CSEC Biology, the cover page. I am Mr. Wilson, and today we are looking at the mark scheme for the CSEC Biology SBA. And of course, by now you would have been aware that once you're doing a SBA, a mark scheme is used to mark the lab. The mark scheme pretty much shows for what the grade will be allotted. So there are some skills that you need to master in order to score full marks. Now I'll be showing you on screen what the mark scheme look like. It is pretty much a table, and this table could be prepared in any program. This one has been prepared in Microsoft Word, and there we have it. The skill is pretty much marked out of 10, and once you would have mastered the skills here, of course you'll be scoring 10 out of 10. So here we have it. I'm going to be magnifying the table so that we can talk a little bit more about the skills here. So let's look at the top of the table. Here we have the topic being diffusion and a number beside diffusion there representing the number of labs that we have assessed for diffusion. So of course this is the second lab that we're assessing for diffusion, and it is of course measurement and manipulation that is represented using Now the aim is to determine how temperature affects the rate of diffusion, and pretty much that's the same aim we'll use in the assessment before. Now if you look at the headings here, we have criteria, and those are pretty much the skills to be mastered. Now the marks allotted for the skills is pretty much in a column, and you can see it there. And if you were supposed to master this, then you'd get full marks that your teacher would record on the marks earned. Then there is of course the moderator mark, and the moderator is the individual from CXC, and the CSEC individual has the authority to increase or decrease the score based on their finding. In most cases the score is not the changed. However, the moderator will of course observe you carrying out the particular practical. For it's one of those skills that the teacher will have to see you doing this skill or carrying out the skill. So make sure that you practice, that on the day you will be able to show yourself as being approved. Now the first thing we want to do for this particular skill, so we want to note the calibration of the equipment being used, and we want to of course select the appropriate equipment for the experiment. Now another thing we need to do is to stir the water using a glass rod. The thermometer should not be used because the thermometer might very well break and cause injury to you or others, and of course could affect your grade significantly. It's very important to stir the water column just to ensure that the temperature is even throughout the column. You want to take the temperature of the water before the experiment and at the time of the color change, and also it should be taken while the apparatus is pretty much on a flat surface. That is sort of ideal so as to prevent it from turning over. Another thing to master while taking the temperature is to ensure that the bulb is immersed completely in the liquid without touching the container, and of course you're supposed to give it enough time to ensure that the reading that you're getting is a stable reading. We want to say here that the thermal equilibrium has been reached, so there's no more changes to what's happening in the water column. You want to ensure that it's pretty much stable before you take that reading. You are supposed to read the thermometer at eye level, and this should be done while the thermometer bulb is still immersed completely in the liquid. Of course, this is very, very important to ensure you have an accurate reading of the liquid and not a reading of course, the atmosphere. You want to handle the thermometer with care to prevent damage or injury to yourself or others. And at the end of the experiment, you want to ensure that you have that thermometer to use again. So you want to ensure that the apparatus is pretty much properly cleaned and stored after the experiment. Once you would have mastered all these skills, then you would be scoring a big 10 out of 10 for this particular lab. You're expected to do at least carry out two of the skill for the duration of your study, and that would give us a total of 20 marks. Now, I do hope that you are better informed now for your SBA, and of course you'll be scoring full marks finding the temperature of a liquid, so you'll be able to effectively use a thermometer. If you have not yet liked, share, or subscribe, it might just be time for you to do so. And when you subscribe, ensure that you hit that notification bell so that you can be informed as soon as there is a new publication. If you have not yet, check out our experimental skills and of course our skills for biology and the many videos that we have on the channel. You can also join us on a Wednesday, Friday, and of course Sunday at 5:05 p.m. when we stream live from Jamaica topics that are pertinent to the syllabus. Thanks much for watching. Take care.